Hello, everyone, and welcome to today's webinar, sCMOS Cameras for Light-Sheet Microscopy. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Teledyne Photometrics. To learn more, visit photometrics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Professor Ulrich Kubitscheck, Professor of Biophysical Chemistry, University of Bonn, Dr. Abhishek Kumar, Marine Biological Laboratory Investigator and Chan-Zuckerberg Initiative Imaging Scientist, Matthew Kose-Dunn, Content Manager, Teledyne Photometrics, and Phil Allen, Product Manager, Teledyne Photometrics. Professor Kubitscheck, Dr. Kumar, Matthew, and Phil, you may now begin your presentation.
And welcome to another Teledyne Photometrics webinar. This webinar is going to be all about light sheet microscopy, the benefits, the challenges, and how such a variable application as light sheet microscopy can have all its needs met by modern CMOS camera technologies. A little introduction to us here at Teledyne Photometrics. We design and manufacture these high-end cameras for biological and life science imaging. This helps us to support demanding quantitative research with these imaging devices that allow you to have very high image quality but retain all the information in your data. This is mostly scientific CMOS technologies. Some of you may know as Photometrics. Since 2019, it's been Teledyne Photometrics, as we were established as part of the Teledyne imaging. This webinar today involves myself, Dr. Matthew Kose-Dunn. I work at Teledyne Photometrics as an application specialist. We're also going to involve Dr. Phil Allen, who is our product marketing manager, who's going to talk to you later on about some of our product lines that are very suitable for light sheet. But the main focus here is our guest speakers, who are both experts in the field of light sheet microscopy. Firstly, we have Professor Ulrich Kubitscheck at the University of Bonn and Dr. Abhishek Kumar at the Marine Biological Laboratory in Woods Hole, and we'll hear from them both later on. Firstly, an outline of light sheet microscopy. The concept here is to minimize photodamage and bleaching. If your sample is exposed to very high intensities of light over the long term, it can cause a lot of negative effects. Light sheet also avoids out of focus light, often seen in wide field techniques. It can optically section 3D specimens, which is seen also in confocal technologies. This allows you to freely image large 3D specimens. You can also do this at very high speeds, so you can observe very fast dynamic events, and you can image them over the long term, thanks to the minimized photo damage. And due to the open nature of light sheet, there are many different variants that exist and can be modified specifically for your imaging needs. So as an overview, you can call these techniques light sheet fluorescence microscopy, or LSFM. These diagrams here show you how the illumination and detection is different for these different techniques. If we look at wide field, you can see that the entire sample in your field of view is bathed in light. The field is very wide. Everything you're looking at is being illuminated, and this can cause damage and bleaching. It also means you're getting fluorescence emission back from samples that you're not focusing on outside the focal plane, seen here by the horizontal blue line. This means you get out of focus light and all kinds of artifacts. The alternative is confocal, where you're using pinholes. This works to eliminate out of focus light, but with techniques like laser scanning confocal, it's a very high intensity laser dot that can damage your sample, And with techniques like spinning disk confocal, you also have to make sure that your disk speed can match up with the speed you want to image, and there's a number of different things to take into account. And spinning disk microscopy can be very technically challenging. One of the alternatives that's come up is light sheet microscopy. As you can see here, the illumination and detection are now occurring at 90 degrees to each other. The illumination comes in from the side as almost, in this case, an hourglass-shaped light sheet. At the thinnest part of the hourglass, this is where it's intersecting with your fluorophores in the sample, and it's only across the focal plane. This is how it can eliminate out of focus light, and this is how you can also maintain illumination on just a single plane, and this allows you to also minimize the photo damage. So by decoupling the illumination and detection in this manner, as seen in the diagram, light sheet microscopy offers the ability to still image these large samples, but with less light dose. So you illuminate a thin plane of the sample, you get minimized photo damage, optical sectioning, you eliminate the out of focus light, and some of these techniques can also be faster than laser scanning confocal, which allows you to image more dynamic events. This is also known as SPIM, or Selective Plane Illumination Microscopy. The point here is that this is the most basic form of light sheet microscopy, and there are many different variants of SPIM. The objectives can be in all sorts of different places. You may require multiple illumination or detection objectives. This is the basic format that has evolved over the years to meet different needs in imaging. Here are some popular light sheet systems. For all of these, the illumination objective is in blue, and the detection objective is in green, with our sample being the black circle. We have SPIM, we also have T-SPIM. T-SPIM essentially means you're illuminating from both sides of the sample. This can eliminate shadows that may form if you're just imaging from one side. You also have M-SPIM, where you take this concept further and have multiple detection objectives. And then you have different ways of positioning the objectives in order to image different samples and allow yourself to have different sample geometries. You have oblique SPIM, tilted light sheet, reflective light sheet, and you also have inverted and dual view inverted SPIM, where essentially you can now have the sample on a surface and you have the objectives at a specific angle, so you can flip the whole thing upside down. These different configurations can match the different experiments you're trying to do. Something that's common between most of these is that you do have a restricted choice in terms of the objectives that you can use and the space that your sample can occupy. If you want to use a very high NA oil objective, you may not find it compatible with some of these techniques. And if you want to image a very large, cleared sample, it also may not be compatible with some of these techniques. So while there are a lot of variants, there are some shared restrictions. A recent development in light sheet technology is single objective light sheet. By having one objective doing both the illumination and the detection, this allows you to take off the restrictions on your objectives. Now you can use oil objectives, you can get a higher numerical aperture, you can get higher resolution. You can also have different shapes and environments of samples. You can have these things inside closed atmospheres so your cells can be live while you're imaging them, and you can add additional modular hardware to them. This is SOSPIM, where you reflect the light sheet off a surface and with the same objective detect the emitted fluorescence. This is used in microfluidics installed into lab-on-a-chip systems. These allow for very high throughput systems that you can add flow to. You also have oblique plane microscopy, where you can simply shoot out your light sheet at an angle and collect the emitted fluorescence from the sample. This is developed in several directions, and light sheet is an actively, constantly developing area. So there are always more of these coming out. Some of the examples that are developed from oblique plane microscopy include SCAPE from Elizabeth Hillman, which is swept confocally aligned plane excitation, and the snout scope from the Andrew York lab. There's all kinds of different techniques and developments coming out. So light sheet is a very exciting and active area. These all allow you to push your field of view, speed, and resolution of your technique as far as you can go once you pair it with a suitable camera. So depending on what your experiment needs, there's often a light sheet technique that's suitable for you. But you don't have to take my word from it. Let's speak to our experts and see what kind of light sheets they use, what their opinions are about it, where they think the field is going to go in the future, and what kind of cameras they use on their systems.
My name is Ulrich Kubitscheck. I'm a professor for biophysical chemistry here at the University of Bonn. I'm located in the Department of Chemistry, so the general area is chemistry. Although for my education, I'm a physicist. And so since I do science, I also do microscopy, and that's what really directs me to do and what really fascinates me since decades now. I think meanwhile, almost all available types of microscopy, starting out with plain fluorescence microscopy, then going over to confocal microscopy, to photon microscopy, photo bleaching techniques, and then I stepped into single molecule imaging. And then more and more we noted that very often you need specific microscopes to answer specific biological questions. And that's where I am now and what we do now preferentially.
I am Abhishek Kumar. I am MBL investigator and Chan-Zuckerberg imaging scientist at the Marine Biological Laboratory in Woods Hole. And by training, I am a physicist, but I am here at MBL to develop microscopy tools and work with biologists to push that envelope, basically to understand what are the limitations of existing tools and technologies, and basically see where we can play a role or what we can do to develop new tools which will help biologists to address their biological problems. So that's my research interest. So basically, in a nutshell, you can tell that we are trying to combine hardware or optical engineering, software part from computer vision to data analysis to control software, and apply these two together to address some biological problems. So I feel fortunate that I am at the MBL because whatever we are developing, there is immediate application of those tools. But more than that, we are in a unique position where our commercial partners, our researchers at different universities and various places, whatever new tools or technology they are developing, they're willing to bring it here. So we, being at the MBL, get access to all these latest and greatest technologies early on. And that's very enriching personally because I get to learn about new tools and technology. And also I get to see which direction overall the scientific community is going as far as new developments are concerned.
Yeah, we got into light sheet microscopy by using the cylindrical way of illuminating a sample because we noted it's simply an excellent way how to reduce background. And we use that to perform single molecule imaging in extended samples, where it's great to have really a low background, but also simultaneously a fast camera data acquisition, which is, for example, not possible to achieve with confocal microscopy. But then things developed on their own somehow. Yeah? So we did some applications with that and were pretty successful examining, for example, nucleocytoplasmic transport using that technique. But then I was contacted by people from the neuroscience department here in Bonn, which is pretty big and well-known and very productive, because people noted that light sheet microscopy is an excellent technique for studying brain structure, most brain structure in model systems. And so, yeah, just for the fun of it, we began to work with them, and okay, we found out they want to look at really big samples, yeah, extended samples that with a very high resolution simultaneously. And obviously that's a contradiction. And so, I mean, to buy a microscope was not the best option then. And so in a very close connection with them, we started to develop the microscopes they need and that perform for their task optimally.
So like all of us, I mean, we are excited about light sheet because, I mean, certainly it's a very gentle technique for optical imaging. And it also offers fast speed because we want to capture live things, fast dynamics. So it has all these potentials, and that's why we are all excited about this technology. So, but just to give you, because we are interested in doing live imaging, so we want to be as gentle as possible so that we don't perturb the biology or the developmental phenomena we want to capture. So it's very sample dependent and biology dependent, but we try to be as gentle as possible so that without perturbing that phenomena, we can capture long-term live. Fairly, we are using our light sheet microscopes to do live imaging. So like live cells, live embryos, basically we want to start at very early stage, like when they fertilize and watch their development or capture their developmental process. It's all live. I mean, we have looked at all kinds of, starting from C. elegans, shrimp, I mean, shrimp embryo, squid embryo, fly, fish, so axolotl. So we have looked at all kinds of organisms. I mean, that's again, as I told you, that's the benefit of being at the MBL is people bring hydra, all kinds of creatures. Whatever they can capture on our Stony Beach, they will bring it to our lab and say, okay, can we image on a light sheet?
The workhorse still is a very normal Gaussian illumination that we scan, so a digitally scanned light sheet microscope. And because these are so easy to build and to produce, I think meanwhile we built 4 or 5 or even 6 of them, I don't remember. But for the application of studying brain structure over a large field, then we started also trying out different illumination modes like bezel illumination and the bezel grating illumination that was introduced by Erik Betzig. So we built that and tried that, and currently we are really working rather on airy beam illumination, and so this is one of the ongoing projects where we have an airy beam illumination, but as you might know, airy beam illumination also is involved with producing side lobes for the illumination, and this creates artifacts, background artifacts, and we develop currently new techniques how to get rid of this background. But also we have one instrument that we constructed using 2-photon excitation, and this is used for light zebrafish, the development of the neural system of zebrafish that is done over several days, yeah, so for really long periods of time. That is a normal scanned 2-photon light sheet that also involves bezel illumination, because for 2-photon excitation this is especially preferential.
By now there are 50 different variants of light sheet, and as you know, there are probably about 10, 12 commercial products in the market. So my lab, I mean, because I have a history when I was a postdoc with Dr. Harish Rao and at the NIH and Dr. Daniel Colon Ramos at Yale University, I got involved with DISPIM, dual view inverted selective plane illumination microscope. So still we are continuing that. We are trying to add small capabilities or component to that DISPIM. So I have a DISPIM, and my lab is also developing a 2nd generation or 3rd generation, whatever you want to call, of a DISPIM to look at large and extended samples. So basically we are trying to do some small modifications, so that will allow us to basically image large samples. So that's one component. I'm also interested, if you follow literature, there are a lot more work and developments been done in single view light sheet microscopy, specifically follow-up work from Dr. Elizabeth Hillman's group and Dr. Dunsby's group, Andrew York at Calico, Reto Falca at in Texas, and Loic at CGI. So they are kind of expanded this design to capture slightly higher numerical aperture. And that's very promising because it allows us to use the conventional sample mounting. At the same time, we can have access to large numerical aperture for high resolution imaging. So that's another aspect I'm very much interested in. So that's a second aspect in my lab where we are interested in single view light sheet microscope. And the third aspect is to do multi-view. There are many, because at the MBL we have to deal with large embryos or large samples like squid and all. So for that, doing multi-view, two-sided illumination and two-sided detection, that configuration is also appropriate for what we are doing. So in my lab, we are building a variant of that microscope as well. So I would say there are three different light sheet microscopes, what we are interested in, because there is no universal microscope which works for all kinds of samples. So it all depends. Any microscope is good or bad. It depends on our biological problem and kind of sample we would like to image. So that's why we have different systems for different applications.
According to my view, currently still the largest problem is that of the sample preparation and specimen fixation in the microscope. Also, another point is that many light sheet microscopes do not provide the possibility to easily change the optics, the illumination or the detection optics. Illumination may not be so critical because you can regulate the illumination relatively easily by using, as I said, bezel beams or gratings of bezel beams or airy beams and so on. So you have a lot of options on that side. But for the detection, this is a major problem. Many microscope types do not allow to change from 4x to 10x to 40x detection then. Yeah. So this is really something that is important to do on the fly. So I think this also needs for some companies development work. And of course, the next big thing is the data, which still is really a major problem currently. So, I mean, we always continuously we invest in more and greater data storages and faster transfer and go for search for solutions on that side.
So, in my opinion, there are two major challenges. One is the most important one is data handling. A lot of times we can collect data, but we suddenly feel that we are not ready to extract meaningful information from that pile of data. That's certainly one of the challenges, and which it's not me. I mean, I think people in this community, we are all facing this challenge together. The associated challenge is once you have a multi-view imaging, we need to fuse them together in a smart way so that we can get best out of all views. And that's also, that depends on image quality and all. And if you want to have a very automated process, sometimes it requires slightly higher quality images, or it's a little tricky. So I think that's one challenge. The other challenge I would say is a lot of times people get excited that, oh, we have a fancy light sheet, can I image that? But sample mounting is also a challenge. Depending on what configuration, what type of light sheet you have, the sample mounting requirements are different. So again, it's not like a confocal where you somehow put your sample between cover slips and you can mount on any confocal. Here it depends what configuration light sheet system you have, and accordingly you have to tailor your sample mounting. So in many cases when we work with new samples or new organisms, we end up spending a lot of time figuring out or optimizing the sample mounting, which is very specific to the configuration of a light sheet system we are using. So I feel that that's another challenge. As a community, we need to work together and share notes or exchange notes with each other so that we can learn from each other and make progress. So we have a limited choice of lenses you can use in most of these light sheet configurations. So in many cases, we are using, if you are conventional light sheet approach, we are using water dipping lenses. So there your sample mounting is different. And also sample mounting, like if you are aware of Flamingo system or other systems, there you can, you need to embed your sample in a capillary and agros and all. But if you want to do cell biology, you can't do like that. So there lattice light sheet or dye SPIM or OSPIM or this kind of single objective SPIM where you put your samples on a cover slip and you image on that is more well suited, I would say. Yeah. What are our limitations? So conventional light sheet microscopes, we use a Gaussian beam. So if I extend my field of view, my beam is going to diverge more. So what we do is we use low NA beam, and we don't focus it tightly so that it's not converging and diverging very fast. So you extend your kind of depth in that respect, but you sacrifice your light sheet thickness. So it's a kind of trade-off. So that's one limitation. But there are some other workaround where Reto's group and some other groups, they have demonstrated that you can focus it tightly and you do axial scan. You can do axial scan fast. And there, these light sheet modes on these sCMOS cameras are handy because when you are doing axial scan, you can sync the rolling shutter readout with that beam waist to get, but again, you are limited how far you can scan that in axial dimension or along the optic axis of your lens. So I think that's a major bottleneck for us, because if we extend the field of view, suddenly what you realize is part of your sample is seeing thin light sheet, but other part of your sample is looking at very thick light sheet. So image quality across this plane is not uniform or not similar as we would like. So that's one of the challenge. If you sacrifice magnification and numerical aperture, you can use low NA lenses, and there you can make a thicker light sheet and extend your field of view. Like LSPI type of system where they are looking at millimeters or centimeter large samples, but they make 20 micron thick light sheet and extend the field of view. But if I want to do cell biology, then the 20 micron is a wide field. It's not a light sheet anymore. So it's a kind of relative term depending on what type of samples we are looking at, what length scale, what sort of lens or numerical aperture we are using for illumination and detection. And based on that, I mean, this field of view is, I mean, governed by that, in my opinion. What we are going to soon encounter is even we can go much faster. Let's suppose I can go 3 times or 5 times faster, but we need enough photons to capture in that short, time. So the other thing we also need is bright probes. And that's what some other companies and other labs, like a company like Promega, they're trying to develop these bright probes so that even we can go faster, we will have enough signal to capture. So I think it's all going hand in hand. Like speed, large area, high sensitivity, but at the same time, we need those bright probes so that we can get enough signal in that fast acquisition mode.
First, detection system that is capable of really low light level imaging, because, I mean, photons are always limited, whatever labeling technique you use. Yeah, and secondly, in most applications, at least, that we really require large field of views. And of course, then cameras are optimal that have a large sensor and are very sensitive. So this combination is that what we use most often. What we use are scientific sCMOS cameras. This is a standard now since many years. We started out with back illuminated EMCCBs, but we switched to the sCMOS because we started out using them because they provide this light sheet modes that we explored in one of our publications on confocal light sheet microscopy, where we use the fact that you can really shift the readout region of the camera across the chip during illumination, and you can create a line scanning confocal microscope by that way in a very, very simple and straightforward manner, what provides you with improved contrast for imaging and reduces background. And if you look at large samples, your illumination beam always has to be pretty extended, thick, yeah, in all directions. So background is always an issue in that case. Yeah, now we rather go for CMOS cameras because of their chip size, yeah, and also the small size of the pixels. Yeah, they have these 6.45 micrometer size pixels, and then they provide extremely large fields of view. And of course, that is optimal if you want to look at large regions of a sample. And this is also, by the way, the reason why we do experiments with area beam illumination, because that provides a very long and very constant illumination field. On the camera side, I must say, so for the detection side, I'm pretty happy by now, yeah, because the current CMOS cameras, they provide quantum efficiencies that are really close to 1. Yeah? I mean, this is fantastic, yeah, and they really have a low noise. So a lot of progress has been done on that side, and I'm really, really happy about that. Also, the big sensors, as I already said. Yeah, I mean, especially Kinetics is that camera that we need for this kind of applications. And we found that we can reduce the number of frames considerably that we need to cover a large field of view. And that is simply due to the large sensor. And to our surprise, and yeah, the microscope optics is also capable of illuminating the large sensor in a very good manner. So I think the field number of the objective lens is something like 25 in that we use in that case, but still we were able really to get a very excellent image over the complete sensor for the Kinetics camera. So that produced no problem as we might have thought before.
So here I'm going to speak for myself and my lab. I mean, we use, thanks to Photometrics for your light sheet initiative. That's a very good initiative in my opinion, and I'm honored to be part of that. So ideally what we want high quantum efficiency so that we can detect low light. We want fast speed as possible, and we want as big chip as possible. So all three. And that's what camera companies, they're going in that direction, like your 95B. I mean, that's a very good camera for us because it has a large chip, fast frame rate, and also very high quantum efficiency. I mean, camera is leading in a way because once you guys make this progress and you say that, okay, now we can go faster, then other hardware aspects we are trying to follow that, okay, now we can go faster, let's keep up with that. So in a way, camera technology is also leading the development of light sheet.
Thanks again to our guests, Professor Ulrich Kubitscheck and Dr. Abhishek Kumar. Now we're going to hand over to my colleague, Dr. Phil Allen, who's going to take us through some product lines that are suitable for light sheet imaging. Over to you, Phil.
Hello all, I'm Phil Allen, Product Manager for Teledyne Photometrics. I'll be introducing you to several of our cameras today that are particularly appropriate for light sheet imaging. As our speakers have told you, as Matt has introduced you, Light sheet microscopy is an expanding collection of techniques that demand the most of your sample and the optical and electronic systems involved with the goal of providing you with useful insight about your biology. Whether it's single plane, dual plane, single objective, scanning pencil beam, oblique illumination, scape, axially scanned light sheet, or some other light sheet method, Teledyne Photometrics is a camera to get you the results you want. Cameras for light sheet really need to do it all. With the diversity of approaches being used, cameras need to be highly flexible to meet the needs of the science you're all doing. Cameras need to be sensitive with low noise, high quantum efficiency to capture all the photons coming back from the sample and turn them into a useful result. Cameras have to have the speed to keep up with the events researchers want to capture with their light sheet systems. Imaging neuronal activity in a zebrafish or mouse brain, mapping protein localization in fixed and expanded organs require high frame rates, high speeds, and high pixel throughput. Some light sheet methods can take advantage of digital slit confocal approaches where the output of the camera is matched to the position of the illumination line to enhance contrast. All of the cameras presented today utilize our programmable scan mode to enhance axial focus or swept pencil beam sheet generation. I'm going to talk to you about three specific product families: the Kinetics family, the BSI family, and the Iris 15. You may have heard of various or used various of these products. The Kinetics is our newest, best, and we'll delve into why we're so excited by it. It now has two variants, the originally introduced 29 millimeter diagonal field of view and a new 22 millimeter diagonal field of view. And it really provides you with the ultimate flexibility for your light sheet imaging systems. The BSI and the BSI Express are our classic C-mount cameras, 18 millimeter diagonal fields of view, relatively fast, up to frame rates of 100 frames per second. And then the Iris is our classic example of a smaller pixel camera designed for lower magnification, lower NA sampling, often used for large samples like in mesospin. So these are some of the details of the camera families. The Kinetics family and the BSI family both use kind of industry standard 6.5 micron pixels. Obviously, they have different array sizes of the pixels. Both of them are back illuminated cameras, so they are 95% quantum efficiency at their peak. I'll talk more about the camera modes in a second. And again, the Iris 15 goes up to 5K by 3K sensor array with 4.25 micron pixels. These sensors collect different amounts of the field of view that your optical system could put out. Nicely illustrated here with a series of colored boxes. The Kinetics got the largest field of view of any scientific camera out there, 3200 by 3200 6.5 micron pixels for a 29 millimeter diagonal across the box. The Kinetics is a camera we designed to maximize throughput. Throughput comes with two aspects: how much data can you collect from your system, and how fast can you collect that data. So the Kinetics samples all the field of view your imaging system could put together. If you compare it to the Prime BSI or the Iris, it speaks for itself, more than 2.5 times greater area than the classic BSI area. It's a 10 megapixel sensor, 1-to-1 format, industry standard 6.5 micron pixels. Kinetics 22 is a new member of the Kinetics family, which will collect the maximum area that a C-mount can put out. But the most important part to me as a product manager and you as a scientist is flexibility. How many problems can this device solve? And I think Kinetics has the most flexibility of any scientific camera out there. And that kind of relates to these four imaging modes: dynamic range, capturing bright to dim signals in the same frame; sensitivity, low noise at moderately good frame rates; speed, no one else can do 500, just shy of 500 frames per second at the full array of the sensor. That data comes out at 5 gigabytes per second when you image the full frame of the Kinetics. So ideal for imaging fast processes like voltage sensors or calcium transients. The Kinetics now has a new Subelectron mode with a 0.7 electron read noise. This is the lowest noise mode in our camera line. And because of the way we've actually used the conversion gain, it has EMCCD-like contrast. Very low signals from your sample can give you very contrasty, measurable images. So in quick summary, this is the Kinetics: high sensitivity, high speed, high quantum efficiency, large numbers of pixels with high throughputs. The Kinetics 22, real quickly, all that, but slightly smaller field of view for folks who can't take advantage of the 29 millimeter diagonal field of view of the full Kinetics. So it comes up at 2400 by 2400, 6.5 micron pixels. If your imaging system doesn't cover, why collect data that you don't need? It has a total of 5.7 megapixels, same modes as the Kinetics and same frame rates as the Kinetics. BSI and the BSI Express, kind of classic cameras. They also contain our programmable scan mode, which are very appropriate for many light sheet uses. 2048 by 2048, 6 micron pixels, back illuminated, has a 1 electron read noise mode at 40 frames a second. Can do up to a kilohertz for those who are watching fast frame rates, but only at a limited number of just under 200 rows. The iris camera is being used a lot in light sheet, particularly for things like mesospim, classic SPIM with lower magnification and lower numerical aperture imaging systems. So because there's a 4.25 micron pixel, there are two variants, the 15 megapixel and the 9 megapixel. So just about 5,000 by 3,000 in the 5 megapixel and 3K by 3K in the 9. It comes with both PCIe or USB interfaces, unlike our other two cameras, which come with both interfaces. So I've mentioned programmable scan mode. This is a digital approach where you match the readout of the lines of the sensor in the camera to where your illumination band is. We and others find this as a useful way of collecting the tightly focused illumination beam while discarding scattered light that might fall outside the illumination slit. So if you're using actually scanned light sheet from Reto Fioca's lab, or a scanning pencil beam, or scanning 2-photon or Bessel beam to actually create your sheet, programmable scan mode is very helpful to throw away that out of focus scattered light. So there's two ways to control it in our software. You can actually input the delay you want to create a number of lines that would be integrated, or you can actually just say, I want so many lines. They both effectively delay the readout of the sensor so that you get a band of collection versus a full frame of collection that rolls down the sensor as your focused region or your pencil beam scans the sample. So there's 3 directional controls. Down, up, and highlighted here, down, up, which is actually quite useful for those of you using galvos, and I'll explain that in a second. So with down, the illumination in blue is scanning the sample. The emission light is being rescanned onto the sensor. And then, as you can see, the point of the triangle gets illuminated first and arrives first at the camera. The next scan, same thing, different scan indicated in different color, but we're basically scanning top to bottom in down mode. Not surprisingly, the camera outputs top to bottom. Similarly, in up mode, we're going to tell the sensor to read from the bottom of the sensor and go up. So the base of the triangle arrives first, the point arrives last, and what the camera outputs is as you would expect. No one else has implemented down-up mode. And the benefit here is there's no flyback time in this galvo we've got here, because we're going to scan down and then up and then down and up. So what's going to actually arrive at the sensor from your image are a series of inverted images, but the camera knows what it's doing here. So what the actual camera is going to output is going to flip every other image so that you don't have to do that after the fact. Thank you for your time. Have a good day.
Thanks again to Phil for taking us through the Kinetics, Prime BSI, and Iris families of sCMOS cameras for light sheet microscopy. To follow up on this, we have some real-life examples of the Kinetics and Iris cameras being used for light sheet microscopy. Our first customer story is also from Professor Kubitscheck. This application concerns super resolution expansion light sheet, where essentially samples are physically magnified via expansion, and then you can perform light sheet on them and achieve super resolution results. The main takeaway here is that once you have these much larger samples, thanks to the expansion process, you now need to make sure your imaging field is sufficiently large so you're not constantly stitching and tiling different images together and adding lots of time onto your experimentation. In image A, we can see it takes a typical CMOS camera 10 acquisitions to capture this field. Each one of these acquisitions is in multiple colors. You have overlap between each image. The XY needs to scan across. The more images you're taking, the more time it's taking, the more you're exposing your sample and the longer your experiments are taking. It's just not very efficient. With the kinetics, you only need to take 2 acquisitions. This vastly cuts down on the amount of time taken and it allows you to be a lot more efficient, with the speed of the kinetics also allowing you to run at a higher throughput because you can run this experiment faster. It's a very sensitive camera, so you can often use less light dose. And you can see from the pixel array that you can still image at a very high resolution, as we see in the magnified images C and D. So with the kinetics, it really helps you image these very large samples much faster and much more efficiently. Another example of the kinetics being used for light sheet comes from Dr. Leonardo Ciccone. Dr. Ciccone is imaging cleared mouse hearts. So the image here you can see is a clarity cleared mouse heart. Often it would take, again, a good number of different acquisitions in order to capture a very large sample. But by pairing the kinetics with a custom 2x telecentric lens, you can capture a field of view like this in the centimeter range, allowing you to get a whole picture of the heart in just one acquisition. In the white square here, you can see an image. This is the entire mouse heart captured in just one acquisition. The field of illumination is very flat. The frame rates are very high. And again, this really allows you to up the efficiency and the throughput of your experimentation because you can now capture the heart in just one image. There's no need to scan. There's no need for tiling. And again, from the magnified image, you can see this all happens at a very high resolution. So this simplifies your experiments and it also makes them more powerful with the kinetics. Next, we have 2 custom stories for the Prime BSI Express. This camera is a flexible, compact package with a industry standard 6.5 micron pixel and a high speed, making it great for light sheet imaging. Our first story comes from Dr. James Manton from the University of Cambridge, who is using multiple Prime BSI Expresses for subcellular oblique plane microscopy. This being one of the single objective formats of light sheet using the Mr. Snouty objective that allows you to look at single cells and highly sensitive samples at very high speeds. The Prime BSI Express performs really well in this application because it can image at a very high resolution at a high speed while maintaining a high sensitivity thanks to the near perfect quantum efficiency and the low noise modes. This customer story involves the lab of Professor Reto Fjoka. This lab has developed a technique known as axially swept light sheet microscopy. This technique excels in high-resolution 3D imaging of subcellular structures over large volumes. This is applied, in this case, to endothelial cells that form blood vessels inside zebrafish. With the Prime BSI Express and Programmable scan mode, which Phil discussed, you can image using the readout of the camera synchronized to the scan of the light sheet, and it allows you to really maximize your imaging time while maintaining a really large field of view at a high resolution, again at a high sensitivity. Overall, the Prime BSI Express is a compact, flexible, and reliable package for light sheet microscopy. We also have 2 examples of the Iris 15. Due to the very large field of view and the small pixel, the Iris is a great fit for light sheet microscopy. This is Dr. Fabian Voigt with MesoSPIM. So the MesoSPIM is for mesoscale microscopy. This is for very large samples, and it's an open science project aimed at making these light sheet microscopes for very large samples. As you can probably tell, this is a full mouse brain. Again, this is cleared. The mouse brain has been captured within one acquisition on the iris, so the large detector size and the rectangular sensor of the iris really allow you to capture a lot in a field of view at a very high resolution, which allows you to capture fine features, especially in a sample as complex as a brain. And lastly, we have Dr. Jan Huisken. It really speaks for itself. This is a cleared zebrafish, and you're capturing a huge amount of it, again, in one single acquisition with the Iris 15. So the large chip and the small pixel, you can really optimize this for low magnification imaging. And now you can image huge samples like this at very high resolution, again, without zooming and without having to add additional components onto your experimentation. And that just about covers everything we wanted to talk about today. Just to summarize things, light sheet is a highly versatile technique with many different variations. sCMOS cameras, we present different product lines which have large sensors and very high speeds, which allow you to make your experiments higher throughput and more efficient while also simplifying them. Different software components like Programmable scan mode allow you to have advanced readout control. You have the hardware to do the imaging, but you also have the software to do more powerful imaging and have greater control over it, especially pairing it with the illumination. By pairing the readout of the camera with the illumination, you can further cut down on the light dose and really run longer physiological experiments over the long term without damaging your samples. And again, there's the kinetics. With the kinetics, there's just no need to compromise. You get it all. You get the sensitivity, the speed, the field of view, and the high resolution. It's great for light sheet. We also offer the prime BSI and iris lines, which also perform extremely well for light sheet and match to different applications and different samples that you may want to image. But that's all from us today for this webinar. If you have any questions, please let us know, and thank you so much for attending.
Thank you, Professor Kubitscheck, Dr. Kumar, Dr. Kose-Dunn, and Dr. Allen for your informative presentation. We will now start the live Q&A portion of the webinar with Dr. Kose-Dunn. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what software are your cameras compatible with?
Thanks, Marie. So the cameras that we offer and the ones that I spoke about in the presentation are suitable for all the software you'd use with major microscope company software. Also a great deal of open source software. So we like to use Micromanager, and there's other open source software such as LabVIEW. If you want to write your own software and you have quite an advanced custom application, we also offer Python wrappers, and we're compatible with MATLAB and all these other options for really customizing specific components of the camera, like Programmable scan mode, for what you'd need to do for your imaging. These are all available on our website, and if you want to develop them even further yourself, then we have downloadable development kits and all sorts for the camera drivers so that you can match it up to the software that you'd like to use for your imaging.
Great, thank you. Next question. What is the largest sensor camera you offer?
So while we offer a standard 19mm diagonal field of view, which is the 6.5 micron pixel 2K by 2K, we also have cameras like the Kinetics, which go up to 29mm. This is the largest size we offer. And we also have the Kinetics 22, which goes up to 22, as the name suggests, because if you're not going to use an F mount or a T mount, you can't access the full 29mm. If you're going to use a C mount, then you may as well use something that's going to fit to that size, which is the Kinetics 22. If you're not going to use the full field of view anyway, we can also offer image splitters. So you can take the really big frame of something like the Kinetics and split it depending on wavelength. split it depending on focal plane. So you can do simultaneous multi-plane imaging or simultaneous multi-channel imaging, because you can split the camera sensor up into different parts and use them that way.
Thank you. Next question. Do these cameras connect via SDI or CamLink? What protocol do they use? Does it connect with the NVIDIA Tegra system?
So the cameras we discussed today typically have a choice between a USB, which is USB 3.2 Gen 2, so the 10 gigabit per second version, or PCIe. This PCIe connector allows you to connect it straight onto the motherboard using a card, and it allows you to have the maximum data rate you'd need. So the Kinetics in its fast mode with the full frame is, as Phil mentioned, 5 gigabytes per second data rate. And you can do this with the supplied cables, which are all PCIe link cables. As for protocols, we supply the driver for all of our cameras, which is PVCam. And this can always be pushed in one direction or another. So it's using a .NET framework. If you'd like it to use something else, or if you'd like your system to be adaptable with the driver, we have Python wrappers for the driver, and we can modify in this way as well. As for the NVIDIA Tegra system, I'm not 100% sure, but I can always get in touch with engineering, and I'll make sure I can send you a follow-up answer to that later on.
Great, thank you. Next question. What are the advantages of different pixel sizes?
So the Kinetics and the BSI Express both offer a 6.5 micron pixel, and this really specific size of pixel is designed to match best for resolution at 60x magnification. So it depends on your application. If you're going to look at things that are very high magnification, you can match it to a very large pixel size. But with light sheet, sometimes you want to look at things that are very low magnification so you can fit more onto your field of view. This is why something like the Iris 15 has a smaller pixel of around 4 microns, because this will match up nicely to 20x or 40x, and you get that combination of low magnification but high resolution, so you can image these larger samples.
Great, thank you. It looks like we have time for one more question. Is it compatible with scanning light sheet?
So yeah, you do have techniques for the static light sheet, and you will also have techniques like digital scan or axial scan, where the light sheet is often in motion, or you have a galvo mirror. And this is where you can really use the CMOS camera rolling shutter. And by synchronizing the readout of the camera with the rolling of the beam, you can completely minimize any dark time. And this is where things like programmable scan mode come in. You can add delays to the readout, and you can change the direction of the readout, all to ensure that you match things up with your beam and eliminate any dark time.
All right, and it looks like we can squeeze one more question in. Which objectives fulfill the large field of view of the Kinetics camera?
So the Kinetics goes up to 29. If you have a microscope that can go up to 25 or more, then you can use the objectives that come with this microscope. In the lab here, we've had success with Nikon objectives, but we also work with Olympus objectives and Zeiss objectives as well. And you can use these to get a full field of view image, or as large as your imaging system will allow, with, most crucially, flat illumination as well. So even though it's a large field of view, you can still use these objectives to illuminate right out to the edges of the field.
Great. Thank you, Dr. Kose-Dunn. Do you have any final comments for our audience?
I'd just like to say thank you very much for everyone to attending and registering. If you'd like any more information on this subject, photometrics.com has all the information you'd need on the cameras, as well as the techniques. If you want to hear more about light sheets, we have a learn section. If you want to hear more about the Kinetics, we have product pages. And you can also get in contact with us if you'd like to ask us any further questions or inquire about one of our solutions.
Thank you again. Dr. Kose-Dunn, Dr. Kubitscheck, Dr. Kumar, and Dr. Allen for your time today and your presentation. We would also like to thank LabRoots and our sponsor, Teledyne Photometrics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.